And it's my great pleasure to introduce our speaker today, Dr. Talmo Pereira. Talmo received his PhD in neuroscience at the Princeton Neuroscience Institute in 2021, co-advised by Professors Mala Murthy and Joshua Shavitz. And during his PhD research, he used fruit flies to study how the brain integrates sensory information to pattern behavioral sequences. And for that, Talmo developed hardware for measuring animal motion at high resolution, as well as computational methods for analyzing social behaviors using deep learning and computer vision. And since 2021, Talmo is working at the Salk Institute for Biological Studies as Salk Fellow. And in his ongoing work, he's using computer vision and deep learning to study animal behavior, neuroscience, and also plants. So today's topic is quantifying behavior using deep learning, and I'm looking forward to your presentation, Talmo. Talmo, please.
Thank you so much, and thank you for inviting me to this and giving me the opportunity to share a little bit about our work. So I'll apologize for the lack of perkiness. It is at 7 a.m. here in sunny San Diego, but we'll power through it. Feel free to interrupt. We're gonna have a couple of different breaks throughout the presentation. Feel free to, I'll pause for a second after one of them, see if there's any questions. Otherwise, we can chat a little bit at the end. Okay, with that, let's get into it. Okay, so as a motivating observation, kick off this talk, one thing that is true of all forms of life, whether it's a plant growing through soil, a fly grooming its head, or a giraffe walking through the savannah, is that all forms of life evolve the ability to move. Evolution produced pressures on that led to the natural selection of the ability to be able to perceive the environment and respond to it adaptively, therefore giving rise to the nervous system and the brain. So if we want to understand it, understand biology and the nervous system in all its different forms, it will behoove us to have a good way to quantify biological motion. Put in other words, how do we go from images and videos that look like this to the distilled down essential representation of the degrees of freedom that still reflect the underlying biological processes that are giving rise to movement that look like this. So we can do this at a variety of different levels. At the coarsest level, what we call centroid tracking, we're essentially just going to be tracking a single point on the animal. And this suffices to describe some behaviors, like say this fly walking through space, where the details of how they move perhaps don't matter as much as just where they are in this space. At a finer level, if we want to be able to detect some behaviors, like say grooming, or like you can see here, the center of mass of the animal is not changing. Its particular position in the arena or in space doesn't really matter that much. Rather, what we need to be able to capture are the fine scale kinematics of the individual limbs that are moving. And this is what we call single animal pose estimation. And I say single animal because at an even finer level, we need to be able to track all of the body parts of these individual animals with not just one animal, but with multiple, which might be required if we want to be able to characterize something like, say, social behavior. So a motivating question then for, problem then for my lab is, how can we leverage deep learning and computer vision to quantify behavior across all these different levels, where clearly there are different levels that are more appropriate for different kinds of behaviors. So today, we're going to be talking about a couple different topics. First is single animal pose estimation. Then we'll get into some of the early work that we did in that domain. Multi-animal pose tracking, how that differs from pose estimation, and we'll talk a little bit about some of the problems that are introduced when we move to this domain, as well as our recent and ongoing work on sleep, our multi-animal pose tracking framework. And then we'll move on to a few different applications that we have pointed all of these computer vision deep learning software tools in order to better characterize and quantify. So let's start off by talking about single animal pose estimation and how we got into this whole domain. So recall that we start off looking at these videos. So the problem of a single animal pose estimation is essentially to go from these individual images to the track locations and landmarks of the particular body parts or other regions of interest that you might be interested in tracking of your particular animal. The exact location and position of these type of these landmarks doesn't really matter. In fact, as you'll see in many of these videos, we're going to have a few different color schemes for those body parts, but they're essentially just ones that are going to be arbitrarily chosen by, let's say, the experimenter, which might have an English name associated with each one, but for the purposes of the algorithmic basis, it doesn't really matter. So how do we actually go about doing pose estimation, retrieving these landmarks from video? So around 2015 and 2016, some of the early works in human pose estimation began to use deep learning to great effect. For those of you who may be familiar with the whole process of, say, tracking something on a video, the classical methods relied on essentially subtracting the background from the image so that you can identify the foreground pixels from which you would normally then extract something like the centroid or heading or other landmarks of interest from the images. Here we have pretty remarkable results considering there is a dancer moving in and out of the plane of focus of the camera. The camera itself is kind of shaky and moving, and most challenging, they're wearing black clothes against a black background. And nonetheless, these methods were still being able to track these movements quite remarkably well, despite all these technical challenges. So how do they work exactly? Well, essentially, some of the innovations that allowed this to work particularly well was the fact that first, they were powered by these big convolutional neural networks. And if you're not familiar with deep learning, it doesn't really matter. The point is that it's a large, essentially function approximator that can take an image as input and produce as output a set of confidence maps. Confidence maps, you can think of as essentially body part-wise heat maps. This is a spatial probabilistic representation of the location of the body parts, where essentially you have here overlaid on the original image, but normally just essentially, without the original image, the neural network is predicting these heat maps where the brightest pixel corresponds to the location that the body part is most likely to be located in. We can then use this representation to extract and infer the landmark coordinates by just finding the brightest pixel. And this representation was important because it turned out that it serves as a really strong inductive bias, or it takes advantage of a very strong inductive bias of convolutional neural networks, which is their ability to locate spatially contiguous patch-like features. So if it can detect something like what the shape of a wrist might look like, it can do this at multiple different scales and be able to reason about the geometry of the body. Okay, so we thought, why can't we do this then in animals, right? There's nothing special, nothing particular about this approach that really ties it down to the human domain, but yet that's all that they were being used for. So it turns out that for humans, there existed these massive data sets of hundreds of thousands of labeled images with millions of individual subjects within each that were all annotated, meaning somebody went in and clicked the ground truth locations of every wrist and every shoulder and every eye and so forth, until they had this massive data set. This was collected at great expense and through crowdsourcing methods. Whereas anytime we entered a new lab, anytime we had a new species, or even within the same lab, a different imaging setup, we're going to have exactly 0 labeled images. And so this is what we refer to as the labeled data problem, right? How do we bridge this divide when it's simply not going to be tractable for every single lab to go in and generate hundreds of thousands of labeled images? So to that end, we developed a method called LEAP. LEAP stands for Leap Estimate Animal Pose. It's a cute double recursive acronym that is courtesy of Diego Durando, my former undergraduate mentee. And it worked exactly like how the human pose estimation approaches do as well, which is to say that it takes a raw image as input, which gets fed into a convolutional neural network, which then learns to predict a set of confidence maps, or those part heat maps, from which we can then infer the pose. Now, our key intuition was that we reasoned that perhaps rather than scaling up the size of the data set required to train these models, we would instead scale down the models themselves with the premise that if there are fewer parameters to learn in the model, perhaps we would need fewer labeled data as well. And so we designed a custom network architecture, and we put this to the test. And when we varied the number of images we used to train the models, we found that even with as few as 10 labeled images, we were getting fairly reasonable results, where essentially we were getting predictions where the 90% of those predicted locations would fall within the outer circles in these error plots. And by the time that we got to a couple hundred labeled images, these were getting to pretty much perfect accuracy, right? So something like 1,000-fold fewer images than were required to train those human pose estimation models. Now, a byproduct of creating these lightweight network architectures was that they're super fast to train. So this is at a time where it was thought that you needed really big, expensive GPU equipment to train deep neural networks. And we were finding that we could train it in as few as like 15 minutes on a single conventional desktop workstation GPU. We're also able to predict on unlabeled frames at hundreds of frames per second, allowing us to create a human-in-the-loop training workflow. This is a workflow in which essentially we would leverage the speed with which we could train and predict to then generate predictions on unlabeled frames, which would be imported for initialization of the annotations, which would result in less and less time being required to generate new images, because as we progress through this process, after each iteration, the models will be trained on more data. It will become more accurate. It would have fewer mistakes to correct, thereby taking us from 2 minutes to label each image to just a few seconds. And we demonstrated this could work on all sorts of different data types. It wasn't just for fruit flies or for lab data. The approach really was as general as the underlying algorithms that it was powered by. Okay, I'll pause there just for a minute. If there's any questions, we'll give this just like 20 or 30 seconds or so, and then we can kind of move on. Awesome. Okay. All right. Feel free to drop them in the chat as well as we go throughout this. We can pick them up at the end of the section. So as we transition to this next section, the idea is that while this method works for single animals, there are going to be unique challenges when it comes to generalizing this to multiple animals. But this is going to be a necessary prerequisite to studying some of the most interesting classes of behaviors, that of social behaviors, which, probably don't need to tell this audience, which spends, I'm sure, a lot of time looking and watching at animals, that social behaviors are quite diverse and complex, and it's going to require some unique engineering advances. In particular, at the algorithmic level, there's going to be unique challenges with dealing with multiple animals, namely that while it's trivial to extend those aforementioned algorithms to be able to detect multiple heads, multiple tails, it will be challenging to figure out which head goes with which tail. And this extends from the fact that it's not as trivial as simply connecting the closest head to the closest tail, even if we were to try to come up with a whole bunch of rules based on the distances and angles and so forth. And so this is what we call the part grouping problem. And again, it's one that's quite salient in the domain of social behavior, where animals are closely interacting. The second problem is what we call the identity assignment problem. This is this issue where if we do all of this within a single frame, we also need to ensure that we can associate our detected and grouped animals across multiple frames in time. This is going to be necessary in order to create continuous time series of the movements of these individual animals. So to address these, we developed the successor of LEAP called SLEEP, or Social LEAP. And SLEEP is intended to be an end-to-end framework for the entire post-tracking workflow. It takes you all the way from raw videos through to annotation, training your neural networks, applying those networks to your data, pulling it back in for correction, and eventually tracking and proofreading. And so, put in other words, it's intended to marshal the power of deep learning to be able to get you from raw videos through to any number of tracked body parts on any kind of video with any number and type of animal. And this really has been the fruit of several years of labor with lots of very, very talented engineers that have worked with us across the years to build this out into a really full-fledged and mature software framework. And as a result, we've really been observing a pretty steady and accelerating user growth. And so to give you a sense, right, you've already looked at lots of these little videos, but here's just a short gallery of the types of videos that we were targeting when we were really developing the software, right? And you can kind of see that we have quite a few different rodent data sets with mice, with gerbils, and we see them in a few different settings, right? Including a couple of home cage settings as well as an open field setting. Okay. So one of the key advances in SLEEP really is its engineering, or how much work we really put into developing this out into a really mature, full-fledged software, as I mentioned earlier. And one of the advantages of this is that it allowed us to build a no-code required workflow for going through the entire procedure step by step of being able to get your data tracked without actually requiring that you have any programming experience at all. This owes, again, to lots of different under the hood, behind the scenes kind of innovations that we had to come up with to really enable this to, simply just work, right? There's lots that happen behind the scenes to basically be, able to provide a user experience that's similar to other softwares you might be accustomed to using, like PowerPoint or Illustrator and so forth, right? We have all sorts of hotkeys and graphical interface design choices that are all based on trying to make everything as intuitive and seamless as possible. All right. So here's that little workflow zoomed in, and we're just gonna watch this just for about a minute or so, in which we're going to be showing how we pull in our data, we define the body parts that we want to track, we create a little job queue for ourselves to find the frames that we want to label, and then from there everything is pretty much just point and click, right? We can copy out these annotations, we can drag and drop them, we can rotate them, and we can use all these little utilities to make it much faster to actually generate new annotations when you're doing your initial labeling. But after you do so, you're then able to just train everything right from the interface. It'll show you the progress of your training and allow you to then pull in those predictions once again to create that human-in-the-loop workflow. Once those are pulled in, you can then use those to correct those predictions, as well as select a part of the video that you want to track. Boom, does predictions, does the tracking, and keeps track of the identities of our animals. Awesome. All right. Cool. So there's a lot of different pieces to the sleep system, and I won't have time to walk through every little piece here, but I just want to, because essentially every one of these boxes has a story associated with it. But for today, I just want to focus on one of these little pieces, which is the work that we put into the inference part, or the part that involves predicting things in real time and at high performance. So one of the advances of really putting a lot of work into how we do predictions once we have trained models in sleep is that we can do really fast high throughput offline tracking, meaning where our goal is just process as much data as we can as quickly as we can. But another objective that would really just constitute an entirely different mode of operation would be to be able to enable real-time or tracking, because this would allow us to do closed loop. And so when we created our initial implementation, we were able to get this to around 12 milliseconds or so on a single GPU, conventional. And this was quite fast, considering the fact that this was the same model that we were using for offline inference, meaning we're not sacrificing any accuracy to get to this speed. These are 1024 by 1024 pixel images, quite large, high resolution, but one which is representative of the type of resolution that you need in order to capture social behaviors when we have multiple interacting animals in a large enough arena. But after we partnered up with some folks at NVIDIA to implement an even more performant backend, we're able to essentially triple both of these types of speeds, both to get us to really fast offline inference, as well as real-time inference on the order of just 3 milliseconds. And so here's a little demo of what that looks like. Here we have a pair of fruit flies engaged in their courtship ritual, where by experimental design, the female fruit fly is sexually receptive. And so what we're doing here is that we're detecting, by doing the pose tracking, the approach of a male. This is a courtship approach. It's based on the angles and relative geometry of their body parts. We can detect that in real time and use that to optogenetically activate a set of neurons in the female fly brain, causing her to physically reject him. In other words, we're detecting the social behavior of one animal and using that to control the social behavior response of another, demonstrating the potential for these kinds of technologies in terms of enabling new experimental design. And to make this functionality a little bit more accessible, we've partnered with some folks at Bonsai to create a plugin that allows you to just load in a sleep model, run these models in real time as part of your experimental workflow in Bonsai's visual programming language, allowing you to then create experimental workflows that can take advantage of sleep's real-time tracking capabilities for multiple animals. All right. Cool. So where are we going next? Well, one very natural extension of this is to capture that missing dimension, right? There's lots of behaviors that still occur in 3D that we're not able to properly capture when we only have a single 2D view. And so here you can see what that is beginning to look like. And here is another set of visualizations on this, in which over the next few months we'll be releasing a new interface for doing multi-view 3D multi-animal annotation that takes advantage of having multiple synchronized cameras with known calibration and geometry across them. And then this will then take advantage of a recent integration that we've built with AniPose, another widely used open source library for 3D pose estimation, leveraging triangulation. And finally, one of the other new methods that we've been applying this to is towards grounding our pose estimation in biomechanics by leveraging these physics simulators like Mujoko to further constrain our poses and to then leverage our pose tracking in 3D to study how the animals are producing these behaviors in the first place from the perspective of biomechanics and neural control of movement. Okay. So that was our some of the recent work we've been doing then in developing deep learning based methods for animal pose estimation and tracking. Didn't have time to get into some of the algorithmic innovations there, but I'm happy to chat about that towards the end. So in this next section,
there is one question now, sorry.
Okay, yeah, sure.
Maybe you can take it. Sabine, please.
Yeah, thanks a lot. I was just wondering, how do you deal with a problem if animals, crawl over each other and identities might swap?
Yeah. So that's a terrific question that you certainly chat about. So why don't we just jump over to that section, because we have a few different cool approaches for dealing with that. Okay. So one of the challenges when animals are, so let's say that we can reliably recover their poses when they are even on top of each other. One of the challenges that induces the most amount of manual labor for correction is the fact that we need to track their identities. That becomes really hard when they're really crawling on top of each other. Well, in sleep, we solve this in two different ways. One is through temporal association, using a process we call flow shifting, where the idea is that we're using optical flow, classical technique in image processing, computer vision, to predict where the animals are going to go next. And we use that information to then associate these trajectories, these predicted trajectories, with where the animal actually is now, right? So these little ghosts here that are going to be catching up to our flies are essentially predictions that occurred in the past, and that we're using to predict where they're going to be in the future. And this has the advantage that it works out of the box. It works with visually indistinguishable animals, but it does have the trade-off that it does propagate errors over time. And we'll see another example of this in just a little bit. But nonetheless, it still can work quite robustly because we're essentially just modeling and forecasting the motion of these animals. And when we have a whole bunch of points on their bodies, this helps us to create additional certainty that we're looking at the same animal. This contrasts to traditional approaches that only leverage, say, the centroid of the animal, which can be error prone when you have a crossing. The other pose landmarks give you added context, which adds a little bit of robustness to those conditions. But in another approach, and one that is really compatible with doing this in real time, we go the complete opposite direction. We don't model their dynamics at all, but instead we identify them based on their visual appearance, based on a method we call direct classification, in which essentially we modify our deep neural network architectures to now not only predict the poses, but also predict some representation of the probability of each animal detection being of a particular identity class, where an identity class is totally up to you, but in this example here, refers to, say, the male fly versus the female fly. Now, the downside, of course, is that you need to be able to then distinguish between these different classes, meaning male or female, white or dark coat, or perhaps other arbitrary or artificial markers that you can use to identify them based on their appearance. So this method is no longer as sensitive to periods in which we have lower information, such as when animals cross over each other, allowing us to recover from these kinds of errors, right? Where perhaps the pose tracking was bad, perhaps the animals were blurry or going on top of each other. This induces then propagation of these errors. Whereas in the appearance-based method that we just talked about, even if there are issues that occur, they'll be transient and it'll recover because it essentially has no temporal dependencies. And in rodents, we can, yeah, sorry.
Could you mark the testicles in males? So that's something that's really obvious physically to distinguish them from the female. So have 2 more points to mark an animal with.
Yeah, you can really use whatever you want, right? So in the fly example, we used their striping pattern on their abdomen, which is very visible from the top. The model also leverages implicitly the fact that there's a little bit of difference in their sizes. So even if we have some like more grosser or explicit or more explicit features, like say in this case here, they've actually shaved the animals in different patterns. All of these will be able to be taken advantage of by the models.
I guess the testicles are easier to see from the bottom up though, rather than the top down. So maybe you wouldn't be able to see that difference as easily.
Yeah, exactly. I think for rodents, the thing I've seen the most is either shaving, coat colors, or seen folks do things like try to use paint, something
On
on
the tail,
markers on tail.
Yeah.
The issue with the tail is just that it's not super visible all the time. Like they like to sit on it, they like to curl it, and it's just under the bedding. So usually folks will do things like try to paint somewhere in their back or the back of their neck or something like that. But they also don't love the paint, right? So you need to find really a nice, biocompatible paint, and one that shows up under your camera illumination. Shaving tends to work a little bit better, but it does grow in over time. So it really depends on like the nature of your experiment. But we're working on even better methods to add robustness to this particular challenge. Cool. All right. Let's go ahead and skip back ahead here. Okay. So if there's no other questions, I'm going to get into the third part of the talk here in these last few minutes, in which I just want to talk about show some preliminary data on some of the applications that we've been pointing this towards. So one natural application of this technology that's probably relevant to lots of folks in this audience is this idea of doing high throughput phenotyping. And in our case, we've been applying this in a very broad collaboration to essentially develop a very general purpose system for disease phenotyping across all sorts of different models of disease. And to do this, we've been building out these monitoring chambers that they're a little bit bulky, working on miniaturizing them, but that allow us to do 24/7 continuous monitoring and are powered by sleep to do the pose tracking. And the idea is that then we now have our animals in a nice IRD, ARD approved setup that allows us to do continuous monitoring 24/7 for months and potentially even years at a time. And the advantage of these setups is that, obviously we have to modify all sorts of things, like for example, not have the food be exactly on top of the animals, right? So that we have visibility of what they're doing. But one of the advantages is that we're able to now develop our pose tracking models that work really, really well and have them be extremely robust to all sorts of different errors, but that occur in these images, because we're now in full control of the imaging settings and conditions, and we're able to reproduce that across many, many different setups. And so in that photo, you saw one set of about 4 of these home cage monitoring boxes, but we're in the process of expanding that to a few dozen, eventually getting to hopefully about 100 or so. Again, recording at 50 frames per second, 1280 by 1024 pixel resolution, which is around 2 to 2.5 pixels per millimeter for these animals, which is great to resolve all body parts in rodents. And we're able to track these 15 landmarks all around the body of the animal, affording us the resolution to pull out all sorts of interesting phenotypes. And the idea is that, once again, we're going to be using this to pull out different kinds of quantitative phenotypes relating to pain, feeding, social withdrawal, tremors, motor coordination, and so forth. And we've been applying this in particular to amyotrophic lateral sclerosis and different models thereof, as well as things like pancreatic cancer, which affect the animal's metabolism and therefore their feeding behavior, as well as pain, and Alzheimer's disease, and a couple of other different targets. And so, what do we get once we run our animals through this, right? Well, we end up getting these, super high dimensional. Like I said, it's 50 frames per second times, hundreds and hundreds and thousands of hours, right, of their free behavior in their home cage. We now need something that can help us make sense of it. But first, here's just a few kind of examples of the types of behaviors that we see our animals doing in these, and then we want to be able to recover in these home cages, these like customized home cages, right? Well, so one of the easy ways to start to pull out some kind of analytics or interesting quantifications from all these trajectories is just to associate all of these landmark detections with different features of the environment and between the different animals. So, for example, we get a really fairly reliable readout of what the animals are eating by just comparing how long that they're spending with their head key points on the feeder ROI. And we can also track how much they're interacting with all sorts of different parts of the environment, such as the feeder top, the cage rim, the water spout, and different objects, as well as each other, allowing us to pull out these more, let's say, like interpretable and salient time series that you see on the left, which then can be used as input to classifiers to predict what the animals are doing, or as analytics unto themselves. But all right, one of the advantages of having a massive amount of data is that it's now compatible with using approaches that can take advantage of large amounts of high-dimensional data to find patterns in an unsupervised way. And so this, in collaboration with our friends at Harvard, we've been applying key point MoSeq to this data set. This is an algorithm that is able to model the generative process that gives rise to the pose tracking that you see on the left, conditioning it on there being an existence of a set of discrete states, also known as behavioral syllables. So these are essentially, what you see on the right is essentially a set of ethograms in which each color corresponds to a different distinct syllable that key point MoSeq identified in a totally unsupervised way. So we ran through this, and we wanted to ask the simplest possible question that we could as a test in our preliminary data of whether this sufficed to pull out any sort of interesting biological signal that could correlate with disease. And so what we did was we essentially just took the probability of each behavioral syllable being executed late into the disease and subtracted out the probabilities of the same behaviors occurring earlier into the disease. And what we find is, again, this is a model of ALS that has pretty severe phenotypes. So we knew we were going to be able to find something. And indeed, it does check out, because when we look at examples from when of syllables that the animals perform less, there's behaviors, micro behaviors that the animals perform less later into the disease. That includes behaviors like these, essentially this climbing-like behavior that it identified, this circling type behavior, which again makes sense in both cases if the animal is no longer able to coordinate its muscles, or is having a hard time really locomoting and doing these more active behaviors, which is going to occur as they lose their motor neurons. Indeed, this is what we expect to happen. Whereas things that happen more include this type of hunching, idle-like behavior, and this self-grooming type behavior. Once again, another one that we would expect a priori to occur more as it reflects a response to pain and anxiety, right? So put together, this all kind of gives us a pretty strong signature of the disease itself. And so our goal is to now do this, obviously with many more time points and a much finer resolution, with many more animals, but essentially to build out an entire repertoire or distribution or behavioral signature that can serve as itself a biomarker of the progression of the disease, right? And we want to be able to extend this to create essentially the ethome, or the full repertoire of different behaviors that occur at different stages of different diseases in the same kind of setting, thereby creating a sort of database of disease-related behaviors, as well as cataloging the wild-type behaviors that you might expect to find in your standard Black Sixes. Okay. So happy to talk a lot more about that. But in these last few minutes, I just wanted to kind of highlight a couple of the other more creative applications that we and our collaborators have been using sleep to tackle. So one is to understand how people behave in museum-like spaces. So in a collaboration with Tom Albright, Sarah Gepstein, and the Los Angeles County Museum of Art, we've been applying these kinds of approaches at a much larger scale. So not quite a home cage, but essentially the human equivalent of naturalistic behavior capture, people in a museum where we've essentially gone down and set up a whole bunch of different cameras, created a calibration board that allows us to synchronize the different cameras and relate them to each other. And we've been applying our sleep models to track people from this unconventional overhead perspective. We've been leveraging some state-of-the-art techniques from computer vision to create 3D reconstructions of the individual museum pieces. And we've been using that to drive, to create these reconstructions of the kinematics of individual museum goers in the same space as a 3D model with the reconstructions and recreations of the individual pieces, allowing us to now study the behavior of these individuals, not only in a sort of abstract, featureless space, but in the context of their environment themselves. This also allows us to essentially be able to create these types of renderings and visualizations from the perspective of individual exhibit goers, such that we can now infer, for example, the kinds of image statistics that were in their visual field of view when preceding particular behaviors, giving us access to all sorts of cool new sort of psychophysical type questions that we can infer just from free behavior. Okay, and finally, one other very different domain of application, but that works in the same way, is to leverage sleep to then track the kinematics of growing plants. So this is a collaboration with some folks who are biophysicists and were interested in just tracking plant motion across time-lapse images. So different time scale, but same fundamental problem. And we're able to show that we can adapt sleep to essentially to work quite well in the setting, even though there are many parameters that are quite different, right? We have hundreds of images, not hundreds of thousands. We have no rigid articulated skeleton, and we have thinner and longer types of morphologies rather than more kind of localized and self-contained ones. But this worked well, and so when I started my lab here at Salk, I began talking to some folks in the Salk Harnessing Plants initiative who were essentially trying to combat climate change through a very ingenious approach. The idea is to bioengineer crop plants that have deeper roots, more massive roots, and roots that produce more carbon-rich polymers, like suberin, which is a core constituent of cork, like you have in your wine bottles. And the idea is that bioengineering plants that kind of upregulate all the biochemical pathways that induce all these different processes, we're essentially transforming our crop plants into carbon sequestration machines that pull CO2 out of the atmosphere and stick them into the soil, where they will no longer contribute to greenhouse gases, to the greenhouse effect. And so while they had terrific geneticists, what they were missing was a good way to automate and speed up their phenotyping efforts. So that's where we came in. We applied adapted sleep to work in these cylinder imaging systems. So here, we're not tracking them over time, but rather just across different perspectives. And since all of the previous approaches in the literature that are used to do plant phenotyping all rely on image segmentation, we had to develop an entirely new toolkit for extracting these kinds of phenotypes that plant biologists normally look at, things like the lengths and angles and counts of different parts of the plant root system architecture. And as a result, we can now pull out thousands of different traits associated with each plant, given one of these series, which we can then use to train classifiers or to sort out plants that they're screening through by their likelihood of having these different kinds of morphological traits that correlate with enhanced carbon sequestration. All right. So recapping, we talked about some of the early work that we did in single animal pose estimation, including how in general these approaches work. We talked about some of the more recent work that we've done in multi-animal pose tracking, and some of the unique challenges associated with not only creating a robust software, but to also deal with the variability in the types of algorithms that will work in different settings. And then we talked about a few different applications related to how we can leverage this type of phenotypic data to extract new biological insights. And so with that, I just want to thank lots of the folks who contributed to this work, the awesome folks in my lab who really do all the work that I shared with you today, and our funding sources. And of course, thank you guys for your attention. I'm happy to take any questions. Thanks.